the chemical nature of DNA. Before discussing the semi-conservative model and understanding the James Watson Crick model of DNA, we need to understand a few concepts about the DNA itself. What is DNA? The background of it. DNA was discovered by Friedrich Mischer. It was first observed by Friedrich Mischer in the nucleus of in the nucleus of white blood cells, pus cells, and what he observed there was that it was found in the nucleus. Since it was found in nucleus, this structure was named as nuclein by him. Later on, it was found that a nuclein was acidic in nature, and when it was found that nuclein was acidic in nature, it was then named as nucleic acid. Nucleic acid. Now, when the structure of nucleic acid was further identified by P.A. Levesh. P.A. Levesh was a scientist who further gave the structural components of DNA. And what he did was that he actually told three major components of DNA, which number one include the nitrogenous bases, number two was 5 carbon sugar, pentose sugar, and number three was the phosphate group. All right. So these were the three major components which were present in the DNA in the nucleic acid as a whole. But if we talk about DNA particularly, then we have the phosphate group. Then we have the phosphate group as usual, and the pentose sugar is here, deoxyribose sugar, which is lacking one oxygen on the second carbon. Deoxyribose sugar. All right. And now when it comes to its structure, the structure of DNA is in such a manner that the DNA basically contains a deoxyribose sugar, as previously told. In that deoxyribose sugar, we on the first carbon we have the attachment. Sorry, it's not proper. It's not a pentagon. It's rather this shape. All right. So on the first carbon, we have got the attachment of the nitrogenous base on the first carbon. On the second carbon, there is the lack of oxygen. Oxygen is not present, and reason behind oxygen being not present on the second carbon is that oxygen, when oxygen forms a bond with hydrogen, it causes to form hydroxyl group, which is quite reactive, and that hydroxyl group being quite reactive causes to decrease the stability of DNA, due to which DNA can be degraded after a short period of time. Let's say if we take example of RNA, in which ribose sugar is present, in which there is no oxygen group absent in the second carbon of the pentose sugar, they have less stability as compared to DNA, because the RNA molecules are having oxygen on the second carbon, which causes them to degrade after half an hour within the cell. But if we talk about DNA, it is a more stable structure because of lack of oxygen on the second carbon. So for stability, second carbon is used. On the other side, if we talk about third carbon is in such a manner that all other carbons are having their hydroxyl group on the upper hand side, but third carbon is having its hydroxyl group on the lower hand side. OH is over here, right? So on the other side, when it comes to the fourth carbon is having H on the lower hand side, OH on the upper hand side, while on the other side, when it comes to the fifth carbon is having the OH, which forms a chemical bond with the phosphate group, and H on the other hand side. Now, over here, The fifth carbon is attached with the phosphate group. It is attached with the phosphoric acid actually. That's correct word would be phosphoric acid. This is how the structure looks like. OH. This is the actual structure of the phosphoric acid which is attached to the DNA. Alright, so this is the actual structure of the phosphoric acid. This is the actual structure of the phosphoric acid. Now, what actually happens in the structure of DNA is that here the OH, the hydrogen, which is since phosphoric acid is an acid, it causes it has got OH group in its axis, due to which what actually phosphoric acid does is that it causes to donate one of its proton. Sorry, it donates one of its OH group and causes to form take hydrogen group from the fifth carbon, due to which there is a process occurring which is known as condensation, dehydration, synthesis, due to which this OH and H is released out in the form of water, H2O, and rest of the structure which is formed is rest of the structure which is formed rest of the structure which is formed is like that. This is OH group, this is OH. On the bottom side there is OH. On the right hand side we have a bond formed of phosphate group with the fifth carbon, which is known as the phosphodiester bond. We're going to discuss that as well, that how this phosphodiester bond is formed. Now when it comes to the third end of the third carbon is basically having the OH group. Third carbon is basically having the OH group, which is free ended from one side of the DNA, and the phosphate group is free ended from the other side of the DNA. If we talk about the the structure of DNA in the vertical strand, then it is found in such a manner that first strand is 5 prime 5 dash, which indicates that there is a free phosphate group on the upper 5 dash, and the rounded side represents the free hydroxyl group, which is represented by 3 dash. Now if we are supposed to form an opposite strand, then of course we cannot create a parallel strand in order to form the bond of one hydroxyl of one nucleotide with another nucleotide, due to which the other strand needs to be opposite. Hence, the on the opposite side, the opposite strand basically has 3 prime. It starts from 3 prime and goes till the 5 prime. So this is a basic structure. Now, when it comes to the but when it comes to the structure of DNA, it is DNA is a polynucleotide. It is a D polydeoxyribonucleotide, which means it is a polydeoxyribonucleotide, which means it is formed by multiple deoxyribonucleotides, which are given in the form of a pentose sugar, phosphate group, and nitrogenous base attached. So this entire structure is considered as a single nucleotide, which is which is then polymerized in order to form a polydeoxyribonucleotide, which is known as the DNA. Now, when it comes to the nitrogenous bases over here, there are two major classes of nitrogenous bases. First is the purines, and second are the pyrimidines. Purines are larger in size because they contain two rings, and pyrimidines are smaller in size because they contain only a single ring. what actually happens in purine, they have got four nitrogen atoms, and in pyrimidine we have got only two nitrogen atoms. The nitrogen atoms are present on 1, 3, first carbon. On the nitrogen atoms are present on the first dash, third side, first side, seventh side, and on the ninth side. While when it comes to pyrimidine, the nitrogen atoms are only present on the first side and third side, because seventh and ninth sides are not present because of no imidazole ring inside their structure. Purines are basically having a pyrimidine ring, which is six carbon ring. Overall, it is a nine membrane structure. Purine is a nine membrane structure, and pyrimidine is a six membrane structure. Purine is a nine membrane structure, and pyrimidine is a six membrane structure. What actually happens is that, let me just try to draw the structure of this as well. This is a six membrane structure. Alright, so over here we've got a 9 membrane structure in purine and 6 membrane structure in pyrimidine. When it comes to the structure of purine, purine has got two rings, which number one ring is known as pyrimidine ring. Pyrimidine ring is the first ring, which is 6 membrane, and second ring is known as imidazole ring. Imidazole ring is 5 membrane. It is in actual 5 membrane, but since pyrimidine and imidazole ring are attached together, they cause to form an overall 9 membrane structure. When it comes to pyrimidine, it has only got a pyrimidine ring. It has no imidazole ring. Imidazole ring is absent, and due to which it only causes to form a six membrane structure. Alright. So this is first carbon. Nitrogen is present over here. Nitrogen is present over here. On nitrogen is present over here and over here as well. So this is how the structure is actually formed. When it comes to the purines, these are found in equal quantity as pyrimidines, such that the total number of purines which are found are equal to the total number of pyrimidines, which is signified by the law of Chargaff's rule. Uh, when it comes to purines, we have got two purines which are known as adenine and guanine. On the other side, when it comes to pyrimidines, pyrimidines are three, which includes thymine, cytosine, and uracil in case of RNA, not in case of DNA. So what actually happens is that adenine is always pairs with thymine, and guanine always pairs with cytosine, which is basically uh, the nature of complementarity in DNA. This nature is known as complementary nature, complementary base pairing, such that adenine always pairs with thymine and guanine with cytosine, and vice versa as well. Cytosine with thymine and thymine and cytosine with guanine and thymine with adenine. So in that sense, if we talk about the total number of purine, A plus G, total number of purine A plus G is always equal to total number of pyrimidine, thymine plus cytosine. And when we are going to move T plus C, thymine plus cytosine on the other hand side, then what we are going to do is divide A plus G by T plus C. T plus C, and on the other hand side, what we'll have is 1, due to which this equation shows that the ratio of purine to ratio of pyrimidine is equal to 1, or in other words, A plus G is always equal to T plus C, or we can also say the number of purines are always equal to number of pyrimidines, or we can also say it in a manner that the adenine is found in equal amount to thymine, and guanine is found in equal amount to cytosine. This condition is verified by the law of Chargaff's rule, which was further verified by a scientist which was named as Rosalind Franklin, who performed the X-ray diffraction of DNA. Rosalind Franklin and Maurice Wilkins. He was a British biochemist in the lab of which the Rosalind Franklin worked and performed the X-ray diffraction of DNA. When she performed the X-ray diffraction of DNA, they found the exact nature of complementarity between adenine and thymine and guanine and cytosine, due to which they purposed an X-ray diffraction of DNA, where they told that the width of the duplex width of the duplex width of the DNA is around 2 nanometer or 20 angstrom, while the length of one turn is equal to 3.4 nanometer, and the distance between two distance between two adjacent nucleotides is 0.34 nanometer. and in each base pair there are around and sorry and in each turn there are 10 base pairs. Each turn there are 10 base pairs. Likewise, on the other hand side, what they actually showed in that case was that there are 10 base pairs, and on the other hand side there are 10 base pairs there are 10 base pairs there are 10 base pairs. On the other hand side, if we talk about the width of one strand, then it is around 1 nanometer, right? And on the other hand side, if we these were the things which were described at that time by Rosalind Franklin and Maurice Wilkins. But that was again not enough to describe the entire model of DNA. So what happened at that time? James and Watson and Crick, two scientists named as James Watson and Crick, proposed a model of DNA which was known as Watson and Crick model of DNA, where what actually was present in that model was that within that model there were it purposed that DNA is a double helix duplex structure. DNA is a double helix structure which is coil around its position. The width of the duplex is 2 nanometer. The length of the each turn in the duplex is 3.4 nanometer. Each base pair is having 10. Each turn of the duplex is having 10 base pairs in, and actually the number of base pairs each turn vary depending on the type of DNA. If it is a Z DNA, then they are going to be 12 base pairs, and if there is another type of DNA, let's say X DNA, then there's going to be 10 base pairs. So it was purpose on the basis of X DNA in case of the Watson-Crick model of DNA, right? So DNA, so at that moment, this was a Watson-Crick model of DNA, which also said that element there were DNA is a double helix structure. It has a complementary strand due to it has complementary base pairing, due to which the adenine always pairs with thymine and guanine with cytosine. And secondly, what it actually said was that the two strands of DNA run anti-parallel to each other, such as one strand runs 5 prime to 3 prime and the other strand runs 3 prime to 5 prime. These were the two strands which ran anti-parallel to each other. On the other hand side, the DNA strand was also having some important characteristics that it basically said that the sequence of the nucleotide sequence and the number of nucleotides varies from organism to organism, varies from species to species, and even from organism to organism. So the sequence of DNA nucleotides varies from organism to organism, and that is what makes each organism unique from the other organism. So this is what was proposed in Watson-Crick model of DNA, which was also given a Nobel Prize later on. And Once the Watson-Crick model of DNA was purposed, what they actually started to do was understand the concept of replication of DNA as well. Replication or duplication of DNA, which is an autocatalytic process. DNA has a characteristic that it can do its replication by its own, and that characteristic of DNA to do replication by its own is known as autocatalytic characteristic, is that it can autocatalyze its own replication. Now, based on the concept of autocatalysis, as well as on the basis of complementarity, such that one new nitrogenous base pairs with the complement nitrogenous base, the replication DNA model, which was proposed by James Watson and Crick on the basis of James Watson and Francis Crick on the basis of the complementarity, was a semi-conservative model. In first case, when it comes to the semi-conservative model, what actually it says is that the DNA is having two major strands. The parallel strand and the other one is anti-parallel strand. 5 prime to 3 prime is the parallel one, and anti-parallel one is 3 prime to 5 prime. What actually happens is that they purpose that in the semi-conservative model, the DNA causes to in the semi-conservative model the in the semi-conservative model, the two DNA strands are separated, and each DNA strand serves as a template to synthesize a new daughter strand of the DNA strand from the old parental strand. Due to which it showed that using one strand of DNA, there is synthesis of the other daughter strand, due to which when these two strands would be unzipped, the first strand will form another daughter strand by the addition of nucleotides through enzymatic processes, and the other strand will also form another strand will also form due to which the first strand would be having one side of the DNA and the other strand would be having the other side of the DNA. Now let's suppose if there is one strand, the one parent strand causes to form the other daughter strand through the addition of nucleotides and through enzymatic processes, and the other strand would be having the addition of nucleotides and forming a daughter strand. Due to which we can say that within a newly formed replicated strand of a DNA, 50% of the material, 50% of the genetic material is from parent, and 50% of the new strand, 50% of the strand is the daughter strand, due to which 50% of the DNA is daughter, 50% of the DNA is obtained from the previously formed strand, and 50% of it is formed from the other strand, due to which 50% of it is from the from the already formed DNA present in the cell, and the other 50% of the DNA is present in the and other 50% of the DNA is the one which is newly formed with the help of the parental strand, which serves as a template for that DNA. Now, when it comes to the conservative model of DNA, conservative model of DNA is the one in which suggests the idea that DNA as a whole is prepared newly by addition of nucleotides without any role of the older one DNA in preparing the uh, in preparing the newly formed DNA, such that the older DNA does not play a role in the preparing of DNA, such that the nucleotides themselves under enzymatic processes cause to combine with each other, due to which they cause to form DNA, which was not supportable at that time. On the other side, if you talk about another model, which was a dispersive model, what dispersive model said was that the DNA molecules, the DNA strands are dispersed. They spread out, they disperse, and cause to combine with each other randomly to form a new strand of old and new DNA nucleotides. Due to which this model was also not proved because there was no such evidence on it. Due to which the model which actually fitted in was semi-conservative model. Now, when it comes to the semi-conservative model, it needed to be proved by some scientists in order to get further knowledge into it. So the first scientist who performed this experiment was Michelson and Stahl. Michelson and Stahl's experiment. What actually happened in Michelson and Stahl's experiment was that in the Michelson and Stahl experiment, they utilized a bacterium which was known as E. coli, and that E. coli bacterium which was utilized was having was prepared in a culture which was having a heavy isotope, not a radioisotope, heavy isotope named as nitrogen 15. And that nitrogen 15 became part of the nitrogenous base of the nucleotides of the polydeoxyribonucleotides of DNA. So when the E. coli culture was prepared at the first end, what the actual E. coli culture was prepared firstly. When the E. coli culture was prepared in the N15 containing medium, what they actually saw was that the both of the DNA strands were became heavy DNA, became death of and heavy DNA, because both strands were having N15 and N15, due to which the DNA's density increased, due to which the entire DNA duplex was considered as a heavy DNA because of N15 in both of these strands, right? N15 in both of the strands, right? Because of N15 in both of these strands. And what they actually used was at that point, they actually utilized what they actually utilized at that point was that they utilized a centrifugation machine. But it was not a normal centrifugation machine because it consisted of cesium chloride, cesium chloride gradient. And within the cesium chloride gradient, the chlorine atoms being lighter in size, being smaller in size and lighter in weight, were you know present in the supernatant, and cesium being heavier metal atom was accumulated on the pellet side, due to which the presence of cesium in showed that if a nitrogen is have present in the cesium region, that at that moment it is the heavy nitrogen of the DNA, and if the nitrogen is not found in the cesium region, rather found in the chlorine region, then it is the simple N14 DNA, which is then utilized in the centrifugation machine, right? And this was done by the help of photographs of DNA bands. Of course, when it was viewed through the X-ray diffraction process, what they actually noticed in the X-ray diffraction process was that um, in the first strand in the DNA cesium chloride structure, in the first strand of the DNA, they saw the photographic band of DNA over the side where the cesium atoms were present in the palette. Let me just color it a bit, such that it was present on the side of cesium atoms or at the palette in the first experiment, where there were both N15 chains, and thus the DNA structure was a heavy DNA. In the second structure, we have in the second structure in the second experiment, what they actually did was, in the second replication cycle, in the second experiment, they introduced a normal culture which was having N14 in it, N14 nitrogen in it, due to which the DNA strand which was formed was N15 to N14, which means one strand was N15 nitrogen containing, and the other strand was N14 nitrogen containing. At that moment, when they utilized they this strand was basically known as intermediate density DNA, or we can also call this as a hybrid DNA molecule, which was having heavy strand as well as light strand. Due to which it was hybrid DNA molecule, and when it was centrifuged, it came to know that this DNA strand was present neither on the side of chlorine nor on the side of cesium, but was present on the intermediate side in between the centrifugation machine, in between the centrifuge, which indicated that it was intermediate in nature, right? On the other side, if we talk about the third experiment in which the replication cycle was further continued, they noticed in the third experiment that N15 and N14 were further replicated, due to which the according to the semi-conservative model we have previously studied, N14 would be making a new strand, which would be having N14 as the daughter strand and N14 as a parent strand. The N14 as a daughter strand had the nucleotides obtained from the N14 culture, which is continued with the replication cycle 2, due to which N14 and N14 both being the N14 strand from here causes to form another N14 strand, due to which it causes to form one light DNA, and another N15 strand which is formed in this DNA was utilized to form an N14 strand, daughter strand, due to which it caused to form one light DNA molecule or N14 N14 and another intermediate density DNA molecule which was N15 to N14 or hybrid DNA molecule. At that moment what they actually saw was that there was the when the replication cycle 2 was continued in the replication cycle 2, what they actually saw was that the the DNA of the replication cycle 2 was present further on the lighter side, was present further on the lighter side. And when there was a third replication cycle in which again N14 culture was utilized, what they actually saw was that the N14 strand of the second replication cycle formed N14 N14 DNA, and other N14 strand caused to form another daughter strand of N14, due to which it was formed N14 N14 DNA. N15 strand of one parent strand caused to form another daughter strand of N14, due to which it caused to form one N15 to N14 DNA, which was the hybrid DNA molecule, and the other N14 strand, which was lightweight, another on N14 strand, which worked as a template strand or the parent strand, caused to form the daughter strand, which was also N14, due to which there were three lightweight N14 strands and one intermediate or hybrid DNA strand, hybrid DNA molecule, due to which what we observed here was that the centrifuge was further lightened. The material inside the centrifuge was further lightened, and it was nearest to the chlorine and away farthest from the cesium. At that moment, what they actually saw was that due to the occurrence of due to the decrease of the heavy isotope nitrogen from one replication cycle to another replication cycle, they understood the point that the N14 to N15 understood the point that the DNA replication follows a semi-conservative model in which the prior or already present parent strand serves as a template strand to form the daughter strand, which is going to be formed by the addition of nucleotides and enzymatic activities. Now when it comes to the replication of DNA, we are going to further discuss it, which in the next class hopefully we'll meet again in another video.